Okay, then I think we can make a start. So good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. Now, with this series, we hope to shake up the usual format a little bit by having a mixed format with time dedicated to the presentation, the demonstration, and the discussion of the software toolkit or method presented. Now, typically these sessions will run for about 50 minutes in total, and we will run this series throughout the year on the last Thursday of each month. And you can already see from this slide we're sharing that we've got a great lineup over the next few months, and we're really looking forward to all the upcoming talks. Now, if you're interested in presenting a new method or software you've been developing, please feel free to reach out to us. We'd also love to have you join our series. So before moving on, just a few quick introductions. My name is Lewis Jones, and with me today I have Erin Dillon and William Gertie, and I think Bethany Allen might be around. The four of us are on the development team of Palaeoverse. Now, if you don't know what Palaeoverse is, it's an organization aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. Now, if you want to know more about Palaeoverse, because I won't go into it too much today, you can check out either our GitHub organization and our website, which I believe Will is dropping into the chat right now. So that's all from me for now. Erin will now give you some helpful tips and instructions for this webinar, followed by Will, who will introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. Thanks so much, Lewis. So as Lewis mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 20 minutes at the end dedicated to Q&A and discussion. So during that time, my fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar is being recorded, and it will be made available at the end on the Palaeoverse YouTube channel. However, we will stop this recording prior to the Q&A session in case you prefer not to be recorded. So before we get started, I'd like to really quickly point out a couple of useful Zoom controls at the bottom of your screen. So you'll notice a Q&A button, and you can use this to submit questions to our speaker. And during the Q&A session, we'll either read out your question or invite you to turn on your microphone, and then you can ask our speaker directly. There's also a raise hand button. So alternatively, you can raise your hand and we'll either call on you and invite you to turn on your microphone so you can ask your question. There's a chat function. Currently, you can only chat with us hosts in case of technical difficulties, but we will turn it on to allow you to chat with other attendees during the Q&A period. And then finally, there's a closed captioning button. You can turn that on at any time to view the transcript. So finally, I'd like to highlight a couple of ground rules. Our aim is to cultivate a really open, respectful, and inclusive environment during this lecture series where we can learn from each other. So with this in mind, we ask that you please be respectful and kind with engaging with our speaker and your fellow attendees, particularly when asking questions or interacting with others in the chat. And any form of harassment or inappropriate behavior will be grounds for removal from this webinar platform. So for more information, you can refer to the Palaeoverse Code of Conduct, which can be found on our website, and I believe it is also being put in the chat right now. And with that, I will pass it over to Will to introduce today's speaker.
Thank you, Lewis and Erin, for getting us all up to speed on how this lecture series will work. Today, I have the great honor of introducing the very first speaker of our new lecture series, Dr. Emily Mitchell. Dr. Mitchell is an assistant professor in the Department of Zoology at the University of Cambridge, the curator of non-insect invertebrates at the Museum of Zoology, and a NERC Independent Research Fellow. She is the PI of the Deep Time Ecology Group at the University of Cambridge, and her group is broadly interested in understanding how ecology impacts evolution through deep time from the origins of animals to the present. This has led to research projects investigating the origins of animals during the Ediacaran, applying ecological network models to ancient fossil assemblages, studying modern Antarctic and shallow water benthic community ecology, and even studying how to detect life on other planets. Across all of these projects, Dr. Mitchell actively applies novel quantitative approaches to tackle various paleobiological questions. Today, we are very excited to hear Dr. Mitchell talk about using spatial point process analyses to quantify the behavioral ecology behind trace fossils. Please join me in welcoming Dr. Emily Mitchell.
So thank you, Will, and indeed all of Palaeoverse for inviting me to give this first talk. So the slide's changing. No. Oh, there we go. Bit of light. So the title of this talk is using spatial point process analyses to quantify the behavioural ecology behind trace fossils. And I chose this since trace fossils is probably the most widely applicable fossil group to use these analyses on, because there's so much more of them than, for example, the ediacaran, which I've done a lot of work on. But I will, of course, talk about the spatial point processes more broadly and then finish off with trace fossils. So I wanted to start by just talking very broadly about spatial analyses. So when we think of spatial analyses, different people think of slightly different things, and that generally scales with temporal and spatial scales. So it can be biogeographical, it can include species different distribution models. And of course, we can also use the spatial point process analyses, which is what I've been using a lot of. So this workshop today, we're going to talk about spatial point processes, what they are, examples of different uses, and specifically how they're applied to trace fossils. And at the end, I'm going to run through some code from some trace fossil analyses that we've done. And all the code and examples is available on our website if you want to download and have a look at it yourself. So what indeed is spatial point process analyses? So the basic idea behind them is, well, to start off with, these are methods that were developed originally in forest ecology. And so in the forest literature, there's a huge amount of work that's been done. And this has established really, really crucially what the best models that have known processes and how those then correspond to different spatial patterns. And it's this link between pattern and process that makes these approaches quite so useful for fossil communities. So how they work is you have your specimens, you mark up where they are, and you have, for example, different colors corresponding to different sorts of species. And the idea is that the different sorts of point patterns will correspond to different underlying mechanisms. So if we think of our deep sea corals, for example, these are super spaced out. And the reason they're spaced out is there isn't much food in the deep sea. So if two corals are too close together, there's not enough food for both of them. There's not enough water volume for them to collect enough food. And one of them dies, and that leads to the segregation of spacing out of points. So when we're looking at marking up or working out how to get our point patterns, we can take this wonderful example of sea anemones over 2 kilometers deep in a chemosynthetic community. We can mark up where our little sea anemones are, and then we're left behind with a point map. And we can then go about analysing this point map. So with spatial point process analyses, there are 3 basic patterns: random, aggregated, or segregated. So aggregated, clustered or grouped, segregated, spaced out, thinning, that sort of language is used for these different patterns. So there are many different ways that we can measure how the density of, well, for us, fossils changes across the mapped area. But one that I've been using a lot is pair correlation functions. And the reason I've been using them is they cover very broad spatial scales and also very easy to interpret. So nearest neighbours is more commonly used, I would say, within paleontology, and it's also a distance measure within the spatial point process analysis kind of universe. But nearest neighbour analyses have a couple of little difficulties. So the first one being that you're mapping out the distance from each specimen to the next one, which means that if all the specimens are found within, say, 5 centimetres of each other, it means you have no idea what's going on at 10 centimetres or 20 centimetres. And this limits the sort of scale and complexity of patterns you can find. Whereas something like pair correlation functions or K functions, you go over much, much larger spatial distances. And as a rough approximation, the distance you can describe is approximately half the width of the smallest part of your mapped area. So how do pair correlation functions work? So if we have our points here, the black dots, what you do is what you don't, what the software does is it calculates the density from each point as you increase the radius. So for this point here, the red, it's at 0. As you increase the distance, you count how many fossils, or the relative density, as you increase the radius. And you do this for all of the different points and average it out. And this gives you your pair correlation function. If you have a pair correlation function equal to 1, it means it's perfectly random. If it's above 1, then density is more than you'd expect by random. So if it's 3, then it's 3 times as dense as you'd expect. If it's below 1, then it's segregated or spaced out, so it's less dense. And so this is a way of visualizing it with simulation envelopes. So in order to work out whether we have an interesting pattern, a non-random pattern, for example, we use Monte Carlo simulations. So because the spatial analyses are non-independent, each point will contribute in many different ways to a single measure. It means that they're not strictly statistically significant, so it's more confidence intervals. So if your observed line, your red line, falls well within the grey, that means you have a random pattern. If it's above, then it's aggregated, and below is segregated. But what's really useful is moving beyond these 3 basic classifications and talking about different sorts of patterns. So, for example, if we have our Thomas cluster model, this is a sort of model that corresponds to reproduction. And we can see, whereas different sorts of models are heterogeneous possum models. So, and that corresponds to a background environment. So, for example, if we use our nearest neighbor analyses, we could very much imagine with this sort of model, where we essentially have a patch, an aggregated patch, but everything's random within it. It would be random because you're not getting the larger spatial scale of the whole mapped area. Whereas for this sort of pattern, we would have non-random behavior. But both of these can be clearly articulated with the pair correlation functions. So this is why we use these metrics. And we can also extend beyond these simple models and combine them into clusters of clusters, double Thomas clusters, and then different sorts of combinations of aggregations. So the different processes, the Thomas clusters that we're speaking about, correspond to reproductive processes. So here we have a big parent tree, and you can see all the little babies around it. And so what this is mathematically is clusters where the density within the clusters has structure. It's more dense in the middle, and the density decreases to the side. And this produces a very distinctive pair correlation function. With the environmental or heterogeneous possum models, that's more of an environmental model. So if we think about trying to map out where all the trees are in ocean, obviously trees have to live on land. So the primary pattern you'll be finding is the islands. And then the secondary patterns that you're going to find are the biological ones of the trees. So these have environmental niche models, and we can pick out, using goodness of fit tests, what the most likely underlying process is. So what does this look like? Well, if we go back to our sea anemones, we can fit a random model, and then we can use what is sometimes referred to as a p-value, but it's not a strict p-value. So I refer to it as a PD value because it's not interpreted in the same way. It's interpreted closer to an R² value, in that if you have a PD value equal to 0, you have no model fit, and if it's 1, it's a perfect model fit. So here we can see this is clearly a non-random pattern. So if we're working in the modern, for example, and a lot of our work is in modern benthic communities, we can take environmental measures, so measures of different substrate heterogeneities or chemical compositions, and input that and use that in the models. However, we can't do that so easily with fossil communities. So instead, we approximate this background environmental heterogeneity using a heterogeneous fossil model, and essentially smoothing out where the points are to create this kind of artificial background. Now, when we do this, we can see the model fit is much better. We have a PD value of 0.43, so approximately 43% of this pattern is described by the background environment. If we look at reproductive models, these are the Thomas cluster models, the PD value is higher again, but actually it's then combined together. It's a really good model fit. And this is quite common in the model. You see spatial patterns that are dependent on environmental fluctuations in the background, coupled to reproductive processes. So another thing that's quite common is this small scale segregation. Now this can occur more often than not because things have finite body size. You're not going to often find 2 anemones sitting on top of each other, and so they will have a bare minimum in which they can be next to each other. So how do we go about making our spatial maps? So there are lots of different ways of doing it. If you're going over a small area, you can do photographs. If you have very good contrast, if your fossil's on the background right, you can use photogrammetry. If you're going larger, you can use LiDAR, or you can use high resolution laser scans, which is what we've been doing for our Ediacaran data. So with this particular laser, it goes down to 40 micron resolution in the field. And so you can see this wonderful Ediacaran frond down here. And if we see the video, the laser passes over it, so it doesn't damage the fossils in any way. And then you can see the blue line here. And what this enables, because obviously we can't take these wonderful fossils out of the surface, it would ruin all the community dynamics besides anything else. But we can see this wonderful reconstruction of the surface of the fossil. And here we've got a close-up of it. The grid is 1 centimeter, and we can see the holdfast of the frond. The Ediacarans then were upright in the water column with a stem, and they've got all these wonderful branches that comes off it. And so, yeah, this particular scan is 40 micron resolution, but you can get down to 15 with the latest ones. But all this scanning takes quite a lot of time. So this is a time lapse for an hour of scanning, which covers about 1 meter square. You can see the scanner is mounted on a tripod. It has rubber soles, so it doesn't hurt the fossils. And so using this, we can slowly, through time, collect this wonderful surface data and map out very large areas. So far, we've mapped out about 48 different Ediacaran surfaces, well over 30,000 specimens. And these are kind of what the maps tend to look like. We've got the grey area, which is the area mapped, the different colours, the different dots correspond to different specimens, different colours, different species, and the size of the circle is how high it is up in the water column. And for the Ediacaran, you can see there's a lot of variety in terms of the diversity between the different surfaces. Some are dominated very monospecific by just a single species, others are more diverse. You also have a big size range of fossils. You have little ones as well as really big ones. So this is bed B in Charnwood, and some of the fronds are well over 60 centimetres. But we can use all this data to reconstruct the different sorts of underlying processes. And so for sessile organisms, this is really quite straightforward because there's only a limited amount of processes that can affect sessile organisms. It's how they reproduce, i.e. how they get there, how they associate with their background habitat, how they interact with each other. So these are things like competition and facilitation. And then you also have density-dependent processes. So in the modern benthos, it would be things like coral pathogens and diseases and sponge. On land, it's things like fire. You can also investigate things like the relative distributions of the dead versus alive to get an idea of whether you've got some density-dependent mortality. Because we know that things like diseases and fires spread much, much more quickly in areas of higher density than lower ones. So the example I am going to give to start off with is reproduction from the Ediacaran. This was the first paper I did using spatial point process analyses. I'm focused on this wonderful critter, Fractifusus. So this was from about 565 million years ago. It lived on the seafloor in very deep water. And the reason it was used for this study is just about the data. So for this study, there were 3 surfaces used, and each had over 1,000 specimens on. Now, I think me and my group, we've mapped out probably over 8,000 Fractifusus, which is a huge amount of data, wonderful, wonderful amount of data to be using more quantitative approaches for. So how do we go about investigating their Ediacaran reproduction? So the first thing we have to do is identify where we're seeing reproductive induced aggregations. And this is where the spatial point process analysis come in, because we can distinguish between the ones that are due to a background environment versus the ones that are reproductive by fitting the Thomas clusters or the heterogeneous posons. So and then using this goodness of fit test, the PD value, to pick between them. So what did we find? So for the 3 surfaces we looked at, we've got our pair correlation functions, and we can see that we have, to start with, we have this small scale aggregation at 5 centimeters or so. And this corresponds to the radius of our clusters. On the D surface, we have single. On the E surface, we have the same small scale segregation, but we also have a larger scale segregation. So this is clusters of clusters. So we have our little clusters and then our bigger clusters. And we see the exact same pattern on H14. So this is strongly indicating reproductive events, but because we want to be extra careful, we then look at the sizes, because if they're reproductive, we'd expect smaller ones to be around medium ones and medium ones around larger ones. And this is exactly what we see. We see the large fractal pieces have a random distribution. The medium ones have a single cluster that go around the large ones, and the smallest ones have a double cluster that goes around the single ones. And yeah, and just to confirm, the way we distinguish different sizes was using McClust, very popular package in R, to determine the most likely number of cohorts, which was 3. So again, slightly artificially, we create artificial boundary, but they do so much. And this is what they all look like together. We can see I've got a single randomness, then we've got a single cluster, and then our double cluster. So what's this then look like? Well, what is the mode of reproduction? For sessile benthic creatures, there are 3 main options. You can reproduce via budding or fragmentation. You can reproduce using like coral spawning, releasing propagules into the water, or via stolon or runners. So of the 8,000 or so fractal pieces that we've mapped out, well, there are some fragments or buds. There's nowhere near enough to reproduce the majority of specimens that we're finding. Indeed, when we see the very small ones, they don't look like little fragments. They look like little tiny versions of the larger ones. So we have to distinguish whether we're going for waterborne propagules or via stolons or runners, like your strawberry and spider plants. So in order to distinguish this, we think about what happens when you are releasing propagules into the water. Well, if you're releasing propagules into water, they're far more likely to go with the current, and which means your clusters are going to be oval rather than circular. So we can then look at what's happening with different fractal fuses. And what we can see is that there's a lot of variation depending on the size class. The largest ones are highly directional. They're 4 times more likely to be in one direction than either side. Here, we note the difference of the scale bars. You're seeing variations of only 10 to 20%. So you've got very little directionality. So what this is telling us is the initial colonization phase is actually fractal propagules settling from the water column. They're then reproducing asexually via stolon or runners, and they're doing this twice. So this paper came out in 2015, and I remember chatting to people in conferences and saying, well, you know, what do you think? Do you believe this? And people would be like, well, I'm not so sure. I'm not so sure. I was like, oh, you know, so, you know, what is a problem with the methods? We're like, no, we know the methods are good and you've applied them well. And then be like, oh, you're not sure about the data? Like, no, the data's good. You know, they believe the data and the methods, they just don't quite feel it. And I think the reason is a lot of paleontologists want to be able to see this. And it's a mathematical pattern that's detected. It's not visible on the surfaces. But where is that stolon? And then 5 years later, my colleagues Alex and Frankie Dunn published this wonderful paper, which was actually showing evidence of stolon. So this is the best one. It's quite hard to see if you're not used to looking at Ediacaran fossils, a very low relief. But you can see here it's emphasized. You can see the stolon coming in, attaching to an Ediacaran frond, going along, and then going along again. So you can see them. And Alex and Frankie found 5 different taxa that had these stolon or filaments attached to them across 20 different surfaces. So we had this kind of final fossil proof that the maths was actually correct. So that was very, very exciting. So in terms of reproductive processes, yes, we can find them. We can actually tease about quite complex reproductive modes, but we can also look at different sorts of environmental patterns. So one example that we've worked on is this absolutely amazing crinoid log from the Jurassic in Holzmarden in Germany. It's 18 meters long. It's very hard to get an idea of the scale of it. It's absolutely beautiful. And so you can analyze where the crinoids attach the log, and then look at how the spatial pattern changes along it. And what you can see very clearly is that you can use these heterogeneous fossil models to model, you know, consistent or formulaic changes in density and test theories. So with the crinoid logs, there was this idea of maybe they're floating, or maybe they're kind of in situ on the benthos, and then before they got colonized. And what this study showed is that the way that they attach to the log and the spatial patterns of the log are very similar to sorts of spatial patterns that you see when you're looking for encrusters on boats or whales, which suggests that these logs were actually floating in the water column rather than on the sea floor. Another project done by my master student, Alavia Dongana, was looking at Silurian corals. So here we've got this wonderful Irish in situ coral site, and Alavia is working hard to take some photos and do some photogrammetry. And so for this particular surface, what he found was that if you look at your tabulus and rugose corals, you're seeing a very specific sort of bivariate pattern, which you only really find with facilitation. The rugose, the tabulate corals were clustering around the rugose. The rugose itself were, very, very environmentally driven, but the tabulate ones were clustering around them. So that's very exciting kind of illustration of facilitation. So we get aggregations, we also get segregations in the deep sea. We've talked a little bit about our corals that are super spaced out. We've got our wonderful ophiuroids here. And while these are mobile, what I like about this photo is you can see how they've moved themselves in order to try and maximize the amount of space they have. But the little ones can be close to each other because they need less resources. And we can have different sorts of spatial patterns for the segregations. So we can see the small scale segregation, which could be due to just finite body size, but it could also be due to things like allelopathy and releasing of chemicals. But you can also have patches due to habitat segregation. So if you're picking up a background where the habitat itself is segregated, even if the organism is not, that's one possibility. Or you could have competition occurring. So for example, you have little ones that cluster together, but not the bigger ones, those are spatially segregated. And in the Ediacaran, how this would work is you have the smaller ones coexisting happily, they don't need many resources, so they can be quite close. But as they grow, they need more resources, and so end up segregating. But of course, these patterns are very hard to detect with the naked eye, which is why we need our maths and statistical methods. So another example of kind of detecting these sorts of more subtle spatial patterns was published a few years later. This is looking at 7 different communities and trying to work out the extent to which you find things like competition, reproduction, and background environment. And what we found was quite surprising, that resource competition and the influence of niche-based or environmental processes was actually really limited. So in modern communities, benefit communities, pretty much everything competes with everything else. If you think of a coral reef, it's all about space. You have huge, large percentages of area coverage, often 80 or 90%. And in the deep sea, it's competing for food. They're very, very spaced out. But we only found 4 instances of resource competition across 8 different communities. So to give contrast, because everything pretty much competes with everything else in the modern, the equivalent, given the number of communities in taxa, we'd expect to find is 26. So it's much, much, much more reduced. But we can actually go and detect this, and we can link then this competition to other sorts of morphological factors to try and work out what's driving this competition. Talked a little bit about the modern, and we see a lot more variation in the modern than we do in the Ediacaran. Got this wonderful, wonderful community called the Forest of the Weird, and we did some spatial analyses on this, and we found that pretty much everything was doing a different sort of thing. We had facilitation, we had competition, small and large spatial scales, we had a lot of habitat associations, a lot of dispersal limitation due to clustering. And we also saw differences depending on what part of the substrate it was on. On the areas with faster flowing current, we saw a totally different sort of dynamics with the corals in particular. They changed and started getting a lot more competitive. But I wanted to talk to you mainly about trace fossils because while there are some beautiful in situ communities, fossil communities that we can find, these aren't as common as the trace fossils. So trace fossils are different because they're moving around. It's more about quantifying the patterns we see than about necessarily linking them to the process. And the 2 kind of key ones I'm going to be discussing are burrow tops, so vertical burrows where you see the top, and horizontal trackways. So some work that was done by Hamilton Allport a few years ago in undergrad in earth sciences was looking at the geology, sedimentology, and spatial ecology of these wonderful burrow tops. And he mapped out kind of 4 different surfaces. We're looking at maybe a total of 500-600 different fossils, and to have a look at what the spatial patterns reflect, which is a mixture of nutrient availability, but also how they potentially interact with each other. And what he found is that there was a variety of different sorts of patterns that we're seeing that would have been very hard to detect without these analyses. So we can see small scale segregation, which corresponds about to the diameter of the burrow. But the distance until we start getting aggregation or random behavior does change between the surfaces, which likely reflects kind of how close the other burrows wanted to get next to each other, which could be a function of the substrate and the stability of the substrate, or the amount of nutrients in the seafloor. So when we're moving more onto trails and trackways, so I think very much in an invertebrate sort of way. In theory, you could apply these to the trails and trackways of vertebrates, but you need such huge bedding planes. I think it'd be quite hard to get the numbers needed to do some good analyses. Trying to quantify, I guess, the patterns we're seeing in trace fossils is really hard. And there's been some really fantastic different sorts of approaches. So we have things like the bedding plane bioperturbation index, which quantifies how much you're seeing, how much the substrate's being disturbed. We also have seen some really, really lovely work looking at the patterns of the trackways and trying to work out what sort of optimization has been going on by comparing them to Levi flights, which is suggested for modern living animals to be a way that they optimize their foraging. They're trying to find food, and also Brownian motion, so that's proxy for random walks. We can also use measures of kind of quantifying the heterogeneity by averaging it over the surfaces themselves. So as Will introduced me, and I've been talking about, I've done a lot of work on the Ediacaran, mostly on body fossils, but there is some really interesting and really, really important kind of transitions going on with the Ediacaran into the Cambrian with the trace fossils. So you see the origins of mobility first in the Ediacaran in terms of trackways, and then you see that developing throughout the Cambrian. I can't resist just showing you a few examples of the wonderful sorts of trackways and trails that you do find in the Ediacaran. So you can see these Yorgia trackways. So you see these kind of compressions of microbial mat with the body fossil at the end. You see this Kimberella here with its radial trackways, and some really complex body fossils with bilaterally symmetric organisms from the Schubatan, as well as possible scavenging traces and possible Cnidarian trails from the mistaken point. But the ones I'm going to be talking to you about are the hyperabundant trackways that look like this, again from the Schubatan. So for this study, we considered two different samples. This was a COVID lockdown project. So one of the reviewers asked us why we chose these. And it's like, well, it's the photographs we have on our hard drives, because we can't get out in the field, unfortunately. But we've got this wonderful here from the Schubatan in South China, from the Ediacaran. And then for our Cambrian sample, we wanted something that looked as similar as possible in terms of the sorts of trace makers to the Schubatan. So Xu Hai Zhao, who was with this project, had this wonderful sample from Nagar in southwestern India. And here we've got our Tropicnus-like larger trails and similar smaller trails, as well as trace fossils and some things like Cruziana. So it's quite a nice, well, so they're beautiful, beautiful surfaces to be working on. So in terms of marking up with our trace fossils, to get more in the nitty-gritty of how we go about things, we've got our surface, we zoom in very closely, and we mark up little lines of where everything is. So here we can see a close-up. We've got the different segments for the different lines with different colours in different sorts of taxa. My son walked in on me once when I was working at home. He was like, "Mummy, are you just doing little circles on a photograph? I could do that. Anyone could do that." And there is an aspect to, it is quite monotonous work in many ways, but depending on what you're working with, the identifying of the taxa can be the hard part. And there is, yes, a lot to be working out. The more you mark up, the more you get. And so here we can see the different traces that have been marked up by Scott Evans for this project. And we can see the little dots, which are the midpoints, which is a way to kind of quantify the spatial distribution of these. So the first thing that we can do with these, once we've extracted this information, is extract the lengths and the angles of all the different place fossils. So here we've got some different distributions, just in terms of the small scales and the larger ones. We've also experimented with doing the trails as a single connected one and then lots of little ones to see what difference it makes to our analyses. So for these, we can see some differences within the different patterns. So with the Schubertan, which is this one, we can see very high levels of aggregation. So this is, and you can see the difference between the large trails and then when they're connected. And with the non-connected ones, you can see higher aggregation because they're going to be all part of the same trail. So that kind of makes sense. Whereas in the Nagar, you can see slightly different sorts of patterns going on, and with the arthropod traces. And then if you look between the small and large ones, you can see very, very different spatial patterns occurring. In the Schubertan, it's all very aggregated, whereas they're very spatially segregated in the Cambrian. But the kind of metric that's probably most useful if we're looking at trace fossils is something called the mark correlation function. So a mark correlation function is where you describe the spatial pattern, but each point has a mark associated with it. And so this most commonly is continuous variables that can be angles or lengths. So you can see how the spatial pattern of different trace lengths, for example, varies over the whole surface. So if we have a random mark correlation function, it means that, for example, the lengths are randomly distributed. So there's no essentially kind of behavioural difference on when the trace maker is just choosing to do long or short walks. If they are similar but close, we can see this pattern. Here it's interpreted the other way around to pair correlations, which can be confusing. And if they're different but close, we can see again a different sort of pattern. So what do we find? So what we find for the lengths is very similar between the Schubertan and the Nagar. We see the shorter lengths tend to be close together, and then we end up being random at larger spatial differences. I did try fitting these trace fossils to Levi and Brownian motions, and the model fits are just appalling. It's just nowhere near being well described. But what that's saying more practically is if you're not getting good model fits with the sorts of models that work very well in modern, you are going to have to kind of look at other ways to describe these patterns. The angles are slightly different because in the angles, you don't see any spatial correlation in the Ediacaran, but you do in the Cambrian. So what this is suggesting to us is that we've got these differences of patterns through time. So we're starting off in the most simple case, as your trace maker is walking through a patchy habitat. They're doing essentially a random walk, choosing angles at random, lengths at random, which means they're not going to distinguish where the good quality substrate is. But then over time, when they get to a good part of substrate, maybe they stop walking so quickly, they start making smaller choices to try and stay, smaller lengths of the traces to try and stay within the area. And then the optimal foraging is when they're changing both the lengths of the trails and the angles, so they're turning back to the area, which is really good. So what do we need to do to go about doing these analyses? So we need data, and we need to create a map, we need to mark up where the fossils are and extract it, and analyze this using the R package SpaceStat. So I've uploaded to my website, and hopefully someone will put that in the chat as well, just a small amount of example code and links to the different places where you can download. And Inkscape I use because it's free, same as R. So let's see if we can then, can you still see my screen? Yep.
Yep, looks good, Emily.
Excellent, thanks. So this is using our studio and code. So I was just checking everything around, which more or less does, in terms of how you'd actually go about doing these analyses. So there's a bunch of different packages. that you to load. So extracting the vectors, the markup into R is there's on the website we call it DEX. There's a variety of different options. You need to use a separate one for trails compared to the kind of body fossils, because with the body fossils you can associate many different dimensions with them. So it's not just about their placement, but it's also about their size and the height, whereas the trails are marked off as single lines. So let's open our data. Yeah, right. So we're importing our data. DEX filaments was some R code written by my student Katie Dallahook, which takes your markup and turns it into a nice set of input points. So here we've got this is a sort of file of structure that you need if you've got lots and lots of line segments, so lots of trails. You've got your kind of your start and your end point. So you've got x0 and y0 and x1 and y1. And this is the format that the package we use, spacestat for spatial analyses, likes to see your line segments in. I think more in an x0, y0 sort of way, but that's not how the package spacestat likes it. Let's get the package up there. Well, they're all loaded. So the key ones are these two. So spacestat is for doing the analyses, and then fitting the goodness of fit test is a select SVM. So let's just. So the first thing we have to do is think about what our data looks like. So this is the mapped up data that we want to do. So we know, because we've collected data ourselves, that we didn't map out this entire big area. We only mapped out the area where the trace fossils were, i.e. the rock surface. Now, if we ran the analyses on this whole thing, it would be highly aggregated, but that really isn't useful, because all that's telling us is we only worked on the rock surface. We didn't work on everything in that space. So what we need to do next is create a window. And that's the area around the area that's mapped out. So you can, if you have that information, import the coordinate points of a marked out edge, or, which is slightly more easier to use, you can essentially define a polygon around it. And so this is where you just click kind of outside where the points are. It's good to get close. I've done a lot of sensitivity analysis. You don't have to get super close. So there's a very, relatively common problem that people have when they're doing this for the first time. Because you're interacting with your display, sometimes where you click and see it isn't where it's actually coming out in R. And that tends to be because of your display settings. And if they're not set at the default and 100%, you need to go in there and change it in your default settings. So in terms of things to watch out for, that's one of them. The other thing to watch out for is there are kind of two sets of objects in spacestat. There is your line segments, your PSPs, which is what we're dealing with now, today mostly, and then your point process PPPs, your point processors. So with your line segments, if you have one point outside, it will produce an error when you try to run things. But that's okay for the normal point processors. So it's just something to be aware of to make sure that you've got all your points in that area. So next thing to do is to create your object. So line segments are PSP, and the structure they have is x0, y0, x2, y2. Then you've got the window. Yeah. So that's all fine, and we can then see what it looks like. Very nice little line segment pattern there. So we can also do density plots. So we talked a bit about kind of modeling the background environment. So the way to do this would be to do it as a density plot. And so here, what we've done is essentially there's a background kernel that's smoothing out. The size can vary. You can automate it using this function, which optimizes for being not too broad that you don't capture the pattern, but not too tight so that you're only getting the individual points. So if you're fitting kind of different sorts of models you want to, that's, yeah, a good one to use. So if we want to do some mark correlation functions, we need to move away from the line segments and look at the midpoints, for example, of the angles. And we do that, we can get the lengths as well as the angles for those to create 2-point patterns. And what we can do is then, okay, so that's using a slightly smaller radius, you can see where the midpoints are. Yeah, and we can plot up. Let's go with that. Very simple places and the angles. So this is all, yes, within the spatial environment. It's not actual spatial point process analysis so much, yes. And so what we're doing now is we're going to use this. So let's just give a little bit more detail here. So here, what we're doing is this is what we're looking at, which is the midpoints. We're using the pair correlation function. You can use other metrics if you want. The number of simulations you're running. I'm using 99 here because there's only so much time in the day. For publications, we tend to go to 999. So if you think about running Monte Carlo simulations, the more you run, the greater chance you get outliers, but the mean will get smaller. So you need to kind of get rid of essentially the top and bottom 5%, and that's what the N rank is doing. I don't like their legends. I don't like R's habit of not plotting exactly at 0, so I get rid of those and limiting it so we can see it. So this is a fairly good computer, so it runs quite quickly, and we should be able to see. Yeah. Here we go. So here we've got our Monte Carlo simulation, our random, and our observed data going up and down like this. So when, especially if we're fitting different models, it can be useful to smooth out the kind of essentially the noisy bits a little bit, because if you have if you fit a model that's going kind of bang through the middle of this, it's actually quite a good fit, but the values for it, the goodness of fit test, will be relatively low. So we can do as an alternative a bit of smoothing, and that's done with the Stoyan factor, which for this one I've done quite a high level of smoothing. There, you can see this is very, very, very smooth. Probably over smooth if we're honest. But you can imagine if you're trying to fit lots of different models, you want to get that kind of balance between noisy signals and not. If we want to then compare what it is to random, for example, we can use we can fit the ppm. So the kind of normal analog would be the LM in the R environment. We've got our point pattern. It doesn't work on marked patterns, so you can't have, so that's what you're doing here with the unmarked. You're doing it as random. You can also do trends as heterogeneous Poisson models, so background density models, and you can also then impose using K ppm Thomas cluster models as well. And statistic you use, so that's the model, and then you're simulating it. Sorry, so right, so that's a model fits, and then we can then test how good it is. And so this particular model, we've fit to random 0.38, and you can see that actually it's quite close to random. I mean, we're looking at, maybe 5% variation at some points, but it's still not a fantastic fit, especially here. Something to be aware of is sometimes you get very high fits, but then you look at it and you've got a big aggregation at the start, but then it's random. And so you can then subset the data. So you're only fitting a model to the spatial scales to non-random. If we then move on to the mark correlation functions, similar way, but we're using mark core instead of the pair correlation function, and in this way we can then use with or without the angles. So you can see, so that's how this interval here would be how you essentially restrict what area you're going to. So it's not something that you can really just run and not pay attention to, because it's very easy to just be fitting a lot to random and not realizing that you're fitting a lot to random. This is slightly faster. Yeah. Again, so the save function is true, is one of the things that's necessary in order to be able to then apply the goodness of fit tests. And we can see kind of a difference in what they look like. For the first one, it's a lot closer to random than it is for the lengths that we're seeing. Yes, and that's just doing the same again. So I think I'm a little bit over time. So I am happy to, and yes, I guess the key thing is that the this has the information needed to replicate what I've just seen, in that you've got an example of some data marked up using Inkscape, got Katie's code on her GitHub. If you're doing body fossils or the tops of trackways, there's the code on my GitHub. Use R, it's lovely and free with spaced out, which is wonderful. And the code is downloadable. So yes, very happy to discuss more and answer questions. Thank you.
Thank you, Emily. That was fantastic. We're going to stop the recording